Welcome to Study Time, a televised home learning program produced by Rwanda Education Board.
Welcome viewers, welcome back again to the series of these lessons we've been having on TV, and the process continues and continues. And today we are looking at what the Senior 5s are supposed to be focusing on. Remember, if you're in Senior 6, this becomes an advantage for you to also dive in and be part of the session today. So we are reviewing part of the work we had in the previous lesson, especially things to do with experiments and finding out who did the experiment, what did they get, and stuff like that. But also, as a requirement, do some bit of foundations of research. How do I write an extensive writing? An extensive component of a research-based kind of. So do not get worried about, I'm not at the university so I can do research. It is a requirement, and it's how we grow in finding about a lot of information concerning some of these topics. We need to dig deep, move slightly ahead of the teacher, and be able to get what we otherwise wouldn't have got in class. So we are having the review of interpreting Meselson and Stahl's experiment in DNA replication, and also having to do some extensive writing, like I just mentioned before, about what actually happens during replication. Do we have mistakes? And if they do happen, are they good? How far good can they be? How far worse can they really be? Do we live with them? Do we take advantage of them? Or they are a total mess that sometimes they can result into death? That's what we are going to look at. Now, before we go to these activities, I want us to have a recap of the experiment in which we looked at the process of replication in a semi-conservative way. Remember, semi-conservative. We had three processes: dispersive, semi-conservative, and conservative hypothesis. But our focus, like we said last time, is about semi-conservative hypothesis. So what does come out of this experiment? So from the images we have here, if you can check, as we focus on the slide, that you have three test tubes showing the results on the extreme right side. Nitrogen 15 within the bases and the nucleotides, in which we are looking at 15 as heavier than 14, meaning that you are going to have the lighter bands and the heavier bands. So if we have each growing separately, so 15 growing alone in a culture, 14 growing alone in a culture, what would we actually have? So you would have a lighter band for 14, lighter DNA 14, and then we'll also have the heavy DNA as 15. Transferring E. coli in a medium is a form of bacteria that was used to carry out the experiment of replication. But we also have to think and ask ourselves, why couldn't they use others? At what rate does bacterium reproduce so that we can have generations and generations much faster? Do we need to remember things to do with 30 minutes per generation? So how much longer do we have for the experiment? When these COVID tests come up, how much longer do they take? 24 hours? 2 days? Why is this PCR reaction taking much longer? Why can't we get rapid tests and then we have the results out? Do they have some errors? Do they have some dangers? Why do people still have to go through the polymerase chain reaction? That takes time, but perfect results, less errors. So here we go, transferred in the medium, and we now have a hybrid, the mix of the two, as you can check. And then, when we look at the distribution, if we look at the hybrid, the lighter band, and we move this way down to the heavier component, then we would have 3 sections: lighter, intermediate, and heavier. So the hybrid, therefore, would be a mix of the lighter and the heavier one, forming a hybrid, as we do have across here. One new strand with the old one. So it's a mix of the 15 and 14, taking us around midway of 14.5. Now the question is this exactly what we'd expect to respond to when questions come? Can they be designed in somehow a different way? And if so, how? Remember, you want to beat a terrorist, you must be a second before his thinking. So we'd expect learners as well to beat the teachers by thinking ahead of them. Can you push yourself further? If this is the representation of the experiment, in which way, in terms of assessment, would the question come? So you want a better performance out of yourself in class, then you must think like a teacher. Look at the content. Think about how a question would come in terms of mixing up of the DNA and then getting this intermediate, one old and one new. If one was heavier and another one was lighter, would we have somewhere in between as an intermediate? So if you have that in mind, then it means that you are in position to be able to preset the exam before you actually do it from the teacher. So here we go. Look at the slide again. Remember, it's the previous component we looked at, so it's not new to you. So check through very well and see. Remember, you took the caption of this, and we also looked at a few of the things we needed to kind of focus on. So let's go through this again together. If I look at this amount, and then I go and check this again, what comes to my mind? That at the start of the experiment, generation 0. So time 0. A single band of DNA with a density of 1.730. 1.730 grams per centimeter cubed. Now, if we have the second lining here, the amount has gone down by a certain amount. Those are the two core, the main engine for you to be able to think about the question. Why numbers high? Why numbers low? What's happening between this and this? That after 4 generations, 2 bands were found. Can it take us back to the bands we are looking at? But the main band had a density of 1.7. Remember, we started with this, now here we go. What explanation do we give for the density of the main band that is being identified here? Over 4 generations. The marks are 2. You give a reasoned explanation for that difference. 2, that after 1 generation, only one DNA band appears, but the density has changed. 1. Are you able to make an estimation? When we look at these figures, when we talk of estimations, we do not want precise, exact figures. We are looking at a rough representation of the expected result. So it means that here the answers are variable. The figures are not precise. They are not fixed. So, yeah, slightly above, slightly below, any number, but within that range of the expected answer. Estimate the density of the band. If If it is in figures, you can slide. So you'll find that the answers have some bit of ranges. One can say, I think it's around 1.71. Another say 1.72. Another says, I think it could be 1.73. Within the same range, acceptable data. Because you are making an estimate. So there is no so much precision that is needed. Which, if any, mechanisms of DNA replication are falsified by this result. That takes us to the 3. 1, conservative. 2, semi-conservative. 3, dispersive. So we need to fall back and think of the 3. Which one is represented by this experiment, by this result? And the third component, explaining why the identified mechanisms are falsified. Why do they not agree? Out of the 3, 2 fall out, but why? Do we need to fall back to the definitions of these hypotheses? So that we are able to create a thread that connects the two together. So we're saying, describe the results after 2 generations and which mechanisms, and explain the identified mechanism, if any, are falsified as a consequence. The results of the experiment are saying, no, it's not this, but the other. D, that to be able to describe. So give the way you see the experiment. Give the results the way you see them in the beakers. But, in addition to that, give a reasoned explanation for the results, for the mixtures coming out. From generation 0 times 0, you've just started. And the clock is reading 0.0 seconds. Then after some time, when we reach generation 2, so you have first generation, then the first generation produces another generation, so we call that generation 2. Like we would be saying parents, then their children, and then the grandchildren. That's what we mean in terms of generations. Give this out of 0, then this also gives out of 1 to make 2. Which now takes us to, after the reasoning, we get back to the first one. Explaining why the density of the main band changed over 4 generations. And here we are saying 1, 2, 3, 4, 5 possible explanations for the reasons for the change. 15 being heavier than 14 because of the extra neutron. Remember, masses of atoms are dependent on the core of the atom, in which we have the neutrons. Chemistry, there we fly. So this is where we look at the relevance of chemistry to the subject in biology and how we need to borrow information from chemistry to be able to give a reasoned explanation for what we're seeing now. 2, that generation 0 contained DNA with exclusively, like, no mix. It is 15, it's 15. So forever, we'll always give rise to 15, to 15, to 15. So it's heavier because, after all, we started with an exclusively heavier isotope as a resource to get the chains. 3, that as generations pass by, the part of the lighter one will increase as the mass of DNA doubles because you keep duplicating what you introduced a lighter isotope into. Which takes us back to the other images. More reds coming in and purples kind of disappearing because you're giving it a chance to replicate within itself as it is basing on the other added strand of the red 14. Remember. So, 4 generations pass by and you have containing only N14 isotope. Now, it's dominating and keeps it at a density of 1.7, which means that if we constantly have this, then we can't continue to fall below because we do not have anything less than 14. So we keep at that. A reasoned explanation as to why it keeps at 1.70, lighter than the 15. So it's not up around 1.730 and it's not 1.72 something, but way down, meaning that we are actually having exclusively the red ones, the new strands in certain sections along generations, like we do have here. N15 isotope remains, but combined in strands with 14. That would give us the other one, the in-between, the second band at the density between this and that, meaning that we are making an average. Check that very well. The band between 1730 and 17, somewhere there in the middle, means that you have a mix of the old and the new one. When we go to the next question, check it very well. I'm not reading, so focus on the slides and check. Done the reading? What do we have? That after one generation, only one DNA band appeared, but the density had changed. So we have all that with the calculation now. That's the estimation. Figures may fall somewhere there. So what exactly are they doing? Are we looking at the 1.730? Check that very well. Add to the lighter one, then divide by 2 to get the average, the intermediate, the mix of the two. Remember, heavier, lighter, added together, divide by 2, get the old and the new mixed together. Which, if any, mechanisms of DNA replication are falsified? What does the experiment tell us? That's what we are trying to look at. Are we going semi-conservative? Conservative? If we fall from 1.7 to somewhere in between 1.7 and 1.73 and 1.7, are we somewhere in the middle? Are we having a new and old? Or are we having completely new? Or are we staying in the older component? So we need to look at the results and check which one is kicked out. If conserved, then we would stay. If we started with 15, we would stay with 15. If we started with 14, then we would stay with 14. Again, depending on the generation they ask us. So here we go. Conservative replication. Explaining why the identified mechanisms are falsified. So this is not agreeing with the results. That's what the question is asking us to state. But why? So we need to understand the definition for conservative replication. That for it to be true, then the two bands should appear in all generations after 0. But it's not the case. So it's falsified. It doesn't agree with the results. Checking on the next. C. Remember, we had A, B, C, D. Now we are doing the C part, the third component. Describe the results after 2 generations and which mechanisms, and explain the identified mechanisms. So you describe the results after 2 generations. What do you have? And then you also have the ones that are falsified. And what is it about? Is it as the result of what we have? So 2 bands, one band containing a mixture, 15, 14. Semi-conservative replication. 15 would have to add up with 14 to be able to form a new and an old strand. So the band containing 14 isotopes during replication from generation 1 to generation 2. The new strand consisting of 14, so the lighter one, replicating using N14. So we have 2 bands that are exclusively new
and lighter. So we are just 14. No 15 to be able to go up, so it keeps a bit lighter. In this passive hypothesis, why it is falsified is because here the model is telling us break down, rearrange, all the mixed up with the new ones. Meaning that if we had the 15n and it breaks down, we would have a new 15 with an old 15, but the masses would still remain the same. I hope we are clear here. Check it again before I move it away. Read the question again. Falsifying, we are trying to use the term to be able to give you an idea of how the results are not agreeing with any one or two of the three proposed mechanisms for DNA replication. We are counting down from 5. We should have taken a snapshot of it because the explanation is given. And the last part, are we able to describe and explain the results found in the centrifuging the mixture of the DNA from generation 0 to 2? All the bands, generation 0 containing all the 15, no replication has occurred because we are at time 0. Check that. 0 generation times 0. One band from generation 2 containing a mixture of 15 and 14, a mixture of the two, intermediate, from 1730, then 1700. So somewhere there in the middle for the lighter band. Remember the 3 test tubes, the one in the middle, the hybrid. And one band from generation 2, all replicated DNA containing all the 14, hey, the new ones are now coming up because we gave chance for it to repair up. The new strands consisting of N14 isotopes are replicated using N14. So the nucleotides containing the strands here are having only and only those components. And last, like I promised you, how much do we have as a skill to find out more? Do we have the books to use? How much of the internet do we have at home? Do we know what to look for on the internet? Are you going to copy and paste? Are you going to use your judgment of what is far too wrong and far too correct and you fall somewhere in the middle? Do you respect the sources where you've extracted the information? Those are the things you need in research. How do you value the very people who put this information? How do you make references? Do you know how to make a reference list of the things, of the websites, of the textbooks? Do you give credit and respect and value the information you have used as being put by someone else? Are you waiting to have all these skills when you reach university and you first struggle in your first year? Which takes us to how much further we can actually go. So in this research-based question, this is what I want us to look at. We always hear about genetic diseases. We have friends who have sickle cell anemia. We have friends who can turn their hands upside down and they can bring their legs to the head. Is it a disorder? Are they lacking bones? What about those that have long bones? What exactly happens? So here is the question as we wrap up. That the process of DNA replication, so we check that. Sorry about that. So we change this from if to of. Fix that very well. And there we go. Is controlled by a variety of enzymes that ensure that, oh my God, my scanner should produce an exact copy of my original document. So it needs to be 100% perfect, as in DNA molecules here make an exact copy of that, no mistake. But some of these mistakes go on unnoticed. So we have the error formation rate is estimated to be around 1 base in every 1 million bases incorporated into the DNA. Now, why do we get genetic diseases? Where do they come from? Where are all these errors coming from? So we're saying, however, some mistakes go on unnoticed. So you have a document, you write it, you proofread. Someone reads it again and says, there was a mistake, but it's okay. We are already in the printing room, we can't change it. Newspapers with mistakes, documents with mistakes, but what about us in humans? If I born with this mistake, how far can it go? Do I pass it on? Are these results so devastating? Can they even result into death? Think about it, but don't get scared. Some result into adaptive variability, that we differ and then we end up looking different amongst the species when we inherit these errors. Do we benefit from them? Do they take us to hell so that we die? So questions are, how do you explain the term adaptive variability? I give you a hint, remember that. 2, can we discuss, giving relevant examples, the necessary evil? We are not talking about the devil here. When we talk of necessity, but at the same time evil, we're looking at the good thing and the bad thing out of a mistake that has occurred. If DNA replication is improper, what is the evil in it? What is the necessary? What is the advantage in it? Can we read 12 pages of the document? You can start now, start cooking your head up, have all the sources possible, make your internet ready, look for all the books, and give a lot of examples that you think you can embed in this research question, and write a beautiful, nice document. Maybe it can trigger you to think about going genetics as a form of specialization. So with that, thank you so much for being with us and following up and being able to catch up with the questions and then the responses. And here you go. Do the research and you'll find it interesting. I wish I can share the results with you when you get them. Thank you so much.